Hyb Seq sequencing from NanoString combines the simplicity of NanoString's molecular barcoding technology with single-molecule cyclic hybridization to quickly deliver targeted sequencing results with low error rates. The process begins with the addition of an isolated genomic sample, the target of interest, to the flow cell. Next, the probe pool and fluorescent reporter barcodes are added to the mixture. Each 10-mer probe contains a short sequence that may anneal to the target. The entire probe pool includes many short sequences that together account for every possible sequence permutation. When a given probe anneals to the target, its sequence is then interpreted with barcodes, the reporters, via hybridization. The reporters each contain a specific sequence designed to code for one base. For example, cycle 1 begins with a probe annealing to the target. The bases at the hybridized target are read through a series of 6 fast-exchange hybridizations that produce fluorescent signals. Each fluorescent signal corresponds to the identity of one base. After a base is read, the reporter from spot 1 is washed away, and a new reporter hybridizes to spot 2 to determine the next target base. Once the entirety of the probe sequence is read, the probe is washed away. Sequencing cycles are repeated until all the target regions have been sufficiently covered. The readout from each cycle is binned and mapped directly to a reference sequence. These readouts allow the generation of a multipass consensus to define the sequencing reads for the length of the target region. The Hyb Seq workflow requires no enzymes, amplification, or sequencing libraries. Each cycle is non-destructive and allows for longer read lengths as well as multiple reads on the same native single molecule, drastically reducing error rates. This technology enables the identification and analysis of many different aberrations, such as SNVs, which have been identified as key drivers of mutation in cancer.